Good afternoon. My name's Jamie Findlow, and I'm the scientific lead for meningococcal vaccines at Pfizer. We're really grateful to be given this opportunity today to come and present on the role of genomic data in Pfizer's meningococcal vaccine development. I'd like to start the presentation with a couple of quotations that I've gained from our senior leadership who are unable to be with us here in person today. The first of these is from Paul Balmer, who is our Vice President and lead for meningococcal vaccines, and he states how Pfizer are proud to have been long-term supporters of the Meningitis Research Foundation, MGL, and believe this groundbreaking initiative will continue as an indispensable resource for vaccine development and ultimately advancing public health as these vaccines are implemented and the cases averted. Our second quotation comes from our Chief Scientific Officer, Dr. Louise Jodar, who makes parallels to the COVID-19 pandemic and states how it's recently highlighted the necessity of genomic data for vaccine development and monitoring for the emergence of variants of concern. He goes on to state that the MRF-MGL and the more recent global meningitis genome library have paved the way by demonstrating how data can be collected quickly and freely shared. He states how he is delighted to continue the support of the global meningitis genome library and acknowledge the integral role that this has in meningococcal vaccines and the vaccines for other meningitis-causing pathogens, all of which support the WHO defeated meningitis roadmap. When we look back at the history of meningococcal vaccines, we can see that typing data has become increasingly important. For the development of the plain polysaccharide and the conjugate vaccines that subsequently came, it was serogrouping data that was really important to both their design and also their implementation. And it was because that we were unable to take the same approach of using capsular polysaccharide for MenB vaccines and the requirement to use sub-capsular protein antigens that therefore this data set of these antigens became important for MenB vaccines. Now, although typing data for these subcapsular antigens are now contained within databases such as the MRF-MGL, what you can see from this timeline is that when the MenB vaccines were initially developed, there was no comprehensive data set of these data, and this is a recurring theme that we'll be coming back to throughout this presentation. When we look at what the future holds, well, it really is going to be both these data sets are going to be important and those contained within genome libraries, because with the development of pentavalent ABCWY vaccines, these data are both going to be important for informing vaccine utility. To move back and look at how genomic data can facilitate subcapsular vaccine development, well, there's two ways that it does that. The first is, as we heard before, is antigen discovery. But perhaps more importantly is once you've discovered your vaccine antigen, is evaluating whether it has antigenic potential. And using a comprehensive data set of genomic data, it is possible to evaluate which proportion of isolates have the gene or don't have the gene, what sequence variance there is within those genes, and also if it is predictable from the sequence protein expression. So although these are important aspects that can be determined using genomic data, it's important to mention that there are certain aspects that really need, it can't be done on a genomic level, and usually you need live isolates to do this. So for example, confirming that the protein is surface expressed and accessible to vaccine-induced antibody really does require live isolates. Phenotypic expression, if it's not possible to be predicted from the sequences, you need the strains. And then also the ability to induce the serum bactericidal antibody and measuring its cross-reactivity to different sequence variants. Again, you need these live strains to do that. When we look at what are the key aspects regarding genomic data to appropriately support subcapsular vaccine development, then we think these are the key components. We think that the data set needs to be representative, and representative means different things to different people, and particularly when what is representative for a country does not necessarily mean it's representative for a global basis. The data needs to be detailed and accurate. Any inaccuracies immediately chip away at the accuracy of the data and the perception of those data within it. It needs to be contemporaneous. As we've seen with COVID, it is important to know what's happening now regarding epidemiology in case there have been any changes, so having it up to date is important. The data needs to be accessible. Data hidden behind firewalls is not very good because people can't get the full data sets, so the data should be open access and freely open to all. The data should also be linked to metadata, and that metadata is important to inform analysis. So, for example, whether an isolate is a carriage or a disease isolate is important, but also traceability to actually work out where that strain come in case that live isolate is needed. With the development of Pfizer's MenB vaccine, MenB-Hbp or TrueMember, there have been a number of lessons learned regarding genomic data. TrueMember includes factor H binding protein or FHBP, and this was identified using what's shown on the slide there regarding traditional discovery techniques. And this was whereby different outer membrane fractions were generated. These were used to immunize mice, and the resulting serum were then used in the HSPA assay against diverse strains to look for where killing was achieved. And using this approach, the factor H binding protein was identified as a promising antigen because it did induce a cross-reactive response against those diverse MenB strains. But as soon as this factor H binding protein was identified, a number of questions immediately came up. What proportion of MenB isolates possess the FHBP gene? Which proportion don't? How many different sequence variants are there? What proportion of MenB isolates express the protein? And then pulling all those together, does gene presence, sequence variation, and protein expression vary across meningococcal clones over time or between geographical locations? And then thinking about the immunogenicity analysis, there were questions, well, what strains should be used in the SBA assays? What was required was a representative database of isolates and FHBP sequencing data, but as I mentioned before, that was not available. Initial investigations regarding the development of TrueMember informed on a number of key aspects. The first one, that FHBP sequence diversity did start to be identified, and it was also identified that many different variants actually seem to segregate into two FHBP subfamilies, subfamily A and subfamily B. During development, FHBP lipidation by the addition of a lipid tail to the FHBP antigen, which was there to aid in production, was shown to have an impact on immunogenicity and induced a greater cross-reactive response compared to the non-lipidated antigen. And it was therefore this antigen that went forward into the final formulation. The lipidated FHBP antigens could provide cross-protection against other variants of the same subfamily, but not so much the other subfamily, and therefore to provide broad protection across the full FHBP diversity, it was identified that a subfamily A antigen was required to provide protection against subfamily A strains, and a subfamily B antigen was required to provide protection against subfamily B. The final component was identified, which is not unsurprising, and that is that surface expression was important to allow killing in the HSPA assay, and this was purely to allow antibody to bind. We therefore needed to get an understanding of FHBP diversity, and therefore Pfizer undertook a large study using collecting strains from many different countries over a number of different years to try and get a representative understanding of FHBP diversity. This study, as shown on the screen here, including over 1,800 isolates, was undertaken, and this was to look at FHBP diversity, and the results of that showed and further validated the FHBP subfamily A and B classification. Although many different FHBP variants were identified, then what we also saw is that there were 10 FHBP variants that were predominant, and these accounted for almost 80% of all strains that were collected and evaluated. The next thing that needed to be looked at, because there was no information, was FHBP expression. So a specialized assay, the meningococcal antigen surface expression or measure assay, was undertaken to measure FHBP expression and surface expression alone. The results from that showed that over 96% of isolates surface expressed FHBP, and because the investigation had used strains from many different countries over a period of time, it also demonstrated that expression is stable between those countries and over time. This strain selection was also used to choose strains for the HbSBA assays for investigating immunogenicity, and to truly investigate the breadth of coverage, 4 primary and 10 additional MenB strains, so 14 in total, were identified and used in immunogenicity studies. These strains were representative of FHBP diversity, and to show a true coverage of the true member vaccine, these strains only included those where the FHBP peptides were heterologous to vaccine antigens, and the expression of FHBP on the surface was at low to medium levels. When we have a look what data had been generated in the clinical development of true member, we can see that regarding diversity, it was known that the full diversity had been identified and the segregation into the 2 subfamilies. The predominance of 10 FHBP sequence variants were identified, and FHBP expression was stable between countries and over time. Using the 4 primary, 10 additional, and some outbreak strains, the cross-protection of true member to these strains and different FHBP variants had been shown. However, when it came to individual countries trying to determine the utility of that vaccine, they were generally the same questions posed by all of those. These were such as, what will be the coverage in my country? Are my circulating strains different? What FHBP variants do my country's isolates possess? And do my country's isolates express FHBP? And this is likely to be a continuing theme. And hopefully, once we get these data within these databases going forward, a lot of these questions could be answered in very quick time. To look back at the timeline, this is the development of true member from discovery to licensure, and then just plotting on now the development of the different databases. So we've not mentioned it before, but the FHBP data was on PubMLST, but again, you can see that that was well after the development of vaccine. Same with the MRF-MGL as before, and we've got the global meningitis genome library as previously mentioned too. But looking towards the future, these databases are going to be really, really important and set the basis for vaccine development. They set the basis for implementation and recommendation decisions, but also, as we've seen with COVID, they also can be used for vaccine program monitoring to see what's happening regarding epidemiology and any potential vaccine pressure. So in summary, meningococcal genomic information has become increasingly important and is now an integral component of those 3 aspects: vaccine development, informing the implementation and recommendation decisions, and facilitating the monitoring of vaccine programs, and also the WHO Defeating Meningitis roadmap. The data within a genomic database is required to be representative, current, accurate, available, and have the appropriate supporting metadata. Without that, the utility of the data is reduced. We still require live isolates and therefore different investigations regarding the development of vaccines, such as immunogenicity assessments, surface expression and accessibility investigations. So we mustn't lose the connection to those isolates. And although MenB-Hbp was initially discovered in the absence of FHBP sequencing data, you can see how such data was critical to inform the ongoing development of the vaccine and informs implementations and recommendation decisions. Pfizer are proud to have been a supporter of the genome libraries and see the integral role that they've been in the future. And with that, I'd like to thank you for your time today.